Good morning, everyone. First of all, I would like to thank the Microbiology Consortium of the Philippine Society for Microbiology for inviting me to give this talk this morning. I made a little change in the title of my presentation as I added the word large scale because you will learn later in my presentation that bacteriocins have been utilized in the industry for many years already, although this has remained in small scales. My talk this morning is composed of two parts. For the first part, I will talk about some general information of bacteriocins, and I will give a little focus on circular and leaderless bacteriocins. And for the second part, I will share some of our findings of our two research projects on circular and leaderless bacteriocins, utilizing molecular strategies on how we can facilitate the higher uptake or industrial uptake of these compounds. In particular, I will discuss our work on the construction of inducible and controllable production system of a circular and leaderless bacteriocin that will enable their production using cheaper cultivation media. And lastly, I will share our findings on our work on the bioengineering of the circular bacteriocin Enterocin NKR-5-3B to enhance its bioactivity through NNK scanning. Bacteriocins is a term given to a huge family of antimicrobial peptides that are ribosomally synthesized by some bacterial strains to gain advantage in their ecological niche. These compounds are heat stable and are active in wide pH range. Because of their proteinaceous nature, these compounds are easily degraded by proteolytic enzymes. Bacteriocins do not have any organoleptic properties, meaning they are tasteless, colorless, and odorless. And so, bacteriocins from food-grade lactic acid bacteria are particularly attractive to the food industry as they have been utilized in many food preservation technologies. Bacteriocins are not new compounds. In fact, they were discovered a little earlier than the first antibiotic penicillin. The first report that described the bioactivity of bacteriocins was in 1925. However, unfortunately, the attention to its potential utility failed to materialize due to some unfavorable circumstance. It was only around 1953 that the first bacteriocin nisin was able to reach the market. And despite the approval of many regulatory agencies in major countries and organizations such as the FAO, WHO, the US Food and Drug Administration, the European Union Regulatory Agency, the industrial uptake of bacteriocins has remained limited. Nevertheless, here are some lists of countries that have approved and utilized nisin A as a food preservative. All in all, there are over 50 countries that have approved bacteriocins as a food preservative in different food items. For instance, in Japan, they have been using nisin A to improve the shelf life and overall safety of their cheese, sandwiches, and miso. And here are some examples of other types of bacteriocins that have been used as a natural preservative in different food products, targeting different spoilage and pathogenic microorganisms. Another important possible utility of bacteriocins that have been considered is on its viability as an alternative to antibiotics. This is because these compounds possess a number of positive attributes such as their excellent killing mechanisms that are active even at very diluted concentration. These compounds have lower probability of resistance development because they do not stay long in the environment. Because bacteriocins have different mode of killing mechanisms, they can kill even the dreaded multi-drug resistant pathogens. But perhaps the most promising attribute of bacteriocins is their high amenability to genetic engineering to enhance their potency or specify their type. The consideration of bacteriocins as possible alternative to antibiotics came about because of the problem of antibiotic resistance. Because not very long after the discovery of the first antibiotic penicillin in the early 1930s, antibiotic-resistant strains have emerged due primarily to the unregulated use of antibiotics, particularly in the animal industry. This problem is compounded by the fact that our antibiotic arsenal is rapidly shrinking. There has been no discovery of any new major type of antibiotics in the last 35 years. And according to the World Health Organization, this problem combo is considered as a ticking time bomb that could potentially change the landscape of our present society. According to a study commissioned by the British government in 2014, the problem of antimicrobial resistance will not just lead to millions of deaths per year, but also cause a severe economic impact on the world economy. Many global health experts have said that this problem poses an apocalyptic threat similar to that of climate change. They warned further that if we do not address this problem, we would find ourselves in a healthcare system similar to that in the early 19th century or before the antibiotic era. So clearly, we must find alternative solution or arsenal against these antimicrobial resistant pathogens. And bacteriocins maybe is able to help or supplement our arsenal against these dreaded pathogens. Here I summarize the main differences between bacteriocins and antibiotics. Bacteriocins can be used in both food and clinical settings, but for antibiotics, it's a no-no for it to be used in food. Bacteriocins are primary metabolites, meaning they are synthesized through the ribosome. They are very active even at very diluted concentration. They are very stable. They have no organoleptic properties. They are very amenable to bioengineering. And in general, they have very low to no toxicity to eukaryotic cells. So there are different types of bacteriocins, but in general, they are clustered in two types, the class 1 and class 2 bacteriocins. Class 1 bacteriocins are bacteriocins that contain unusual amino acid residues such as lanthionine and methylanthionine as a result of the enzyme-mediated dehydration reactions of some amino acid residues. For class 2 bacteriocins, they are further subdivided into four categories, the class 2A, 2B, 2C, and 2D. So class 2A are known for their very potent bioactivity against listeria species, including the very pathogenic bacteria and a foodborne pathogen, Listeria monocytogenes. So the next speaker for today's seminar, Dr. Francis Eligado, will discuss more on this type of bacteriocin. But as for my presentation this morning, I will focus on class 2C and class 2D, which are the circular and leaderless bacteriocins. For us to better understand what are circular and leaderless bacteriocins, allow me to first describe how bacteriocins are, in general, are synthesized. The producer strains of bacteriocins synthesize them as an inactive precursor peptide which contain a leader peptide attached to a core peptide. This precursor peptide would undergo enzymatic processing, resulting to the cleavage of the leader peptide, thereby releasing the active form of the bacteriocin. In the case of circular bacteriocin, the active form has a circular structure because the N and C termini of the peptide are covalently bounded by the biosynthetic enzyme cyclase. In the case of leaderless bacteriocin, as the name suggests, there is no leader peptide, which means that this group of bacteriocins are already in its active form soon after translation in the ribosome. Leaderless bacteriocins are one of the most enigmatic group and the most poorly understood group of bacteriocins because leader peptides play a very important role in the biosynthesis of most bacteriocins. First, leader peptides keeps the bacteriocin molecule in its inactive form while it is still in the intracellular space, thereby protecting the producer strain from the toxicity of its own bacteriocin. It also serves as recognition site for the enzymatic processing during its biosynthesis. So here, I show the schematic diagram of the biosynthesis of a leaderless bacteriocin. So it is composed of three systems: the intracellular self-immunity, secretion, and extracellular immunity. Because as soon as the bacteriocin is synthesized from this gene, it needs a protection system that it will not kill the producer strain once it's still inside the cell, and it also needs a system that will pump out this bacteriocin outside of its cell. So once the bacteriocin molecules are already outside the cell, it also needs another system that can protect itself from the toxicity from the extracellular space. So the main actors of my talk this morning are these two bacteriocins, Enterocin NKR-5-3B and LacticinQ. These are two well-characterized bacteriocins. Enterocin NKR-5-3B is a novel circular bacteriocin, while LacticinQ is a novel leaderless bacteriocin. The 3D structures of these bacteriocins and their mode of action are also well studied. But before I proceed, I want to illustrate the promise of circular bacteriocins and also, in a way, leaderless bacteriocins, because later in my presentation I will show that leaderless bacteriocins and circular bacteriocins possess very similar structural conformation. So what we did here is that we compared the stability of different types of bacteriocins against heat treatment using sterilization condition. So we tested the circular bacteriocin, Enterocin NKR-5-3B, nisin, and Bacteriocin. So as shown here, the circular bacteriocin Enterocin NKR-5-3B remained stable even after subjecting it to autoclave condition. But don't get me wrong, these linear bacteriocins are very good antimicrobial agents. As a matter of fact, these two bacteriocins, Nisin A and Bacteriocin PA1, are already commercially available as food preservatives. What I want to emphasize here is that the promise of circular bacteriocins and leaderless bacteriocins far exceeds to that of its linear counterparts. As I mentioned a while ago, it's very interesting to know that circular and leaderless bacteriocins exhibit a common structural fold, or they have a structural resemblance known as Saposin fold. So this Saposin fold is a structural conformation by 4 or 5 amphipathic alpha helices arranged in a distinctly compact architecture that forms a small hydrophobic core. So this is despite the fact that these peptides have no conserved sequence. So this is the amino acid sequence of Saposin, the circular bacteriocin, and leaderless bacteriocin. So as shown here, this is the overlay of their 3D structures. So in showing here that their structures really resemble to each other. If you are interested more on these compounds, I would like to refer you to our paper published in Frontiers in Microbiology last 2018. So here we discuss the molecular details of their biosynthesis, mode of action, applications, and prospects. We also have experimentally verified and validated the genes responsible for the biosynthesis of these bacteriocins, and we have successfully established its heterologous expression system. So in the case of Enterocin NKR-5-3B, the four gene products Ent53B works cooperatively to process the precursor peptide encoded by NIMBB to yield the mature circular bacteriocin. Whereas in the case of LacticinQ, the five genes LnqB-C-D-E-F works for the secretion and immunity of lacticin. So, if these bacteriocins are really good antimicrobial agents, why is it that they are only utilized in the industry in small scale? So the main issue really that is limiting the large-scale utilization of these compounds is on the economics. Because the producer strains of bacteriocins, particularly the lactic acid bacteria, require complex and expensive cultivation media to grow that is making it impractical to use them as food preservative, as it will significantly add to the price of the food product. This is the reason why only rich countries or the advanced countries are able to afford and use bacteriocins as food preservative. And this brings us to the second part of my talk this morning, which is on how we can design ways to improve the industrial utility of these compounds through molecular strategies. And I will be talking now on the Nisin-Controlled expression system for the production of the leaderless Bacteriocin LacticinQ and Enterocin NKR-5-3B, or the NICE system. But what is NICE system? For us to understand this Nisin-Controlled expression system, it is important for me to discuss first the biosynthetic mechanism, how nisin is synthesized by its producer cells. Because NICE system was derived from the nisin biosynthetic machinery. So this is the architecture of the biosynthetic genes of nisin, and it is composed of 11 genes arranged in 3 transcriptional units. And this biosynthetic machinery is grouped into 4 systems: secretion, enzyme cleavage, immunity, and regulation. The genes colored in green encodes for the enzymes responsible for the secretion, the black colored genes for the enzymatic cleavage, the red colored genes for the immunity, and the purple ones for the regulation. So nisin, encoded by the gene A, by the gene Nis-A, is synthesized as an inactive precursor peptide known as Nis-A, and this will be secreted outside of the cell by the enzymes Nis-B, C, and T. It is also during this time that the unusual amino acids are introduced by these enzymes through dehydration reactions. The secreted precursor peptide will then be recognized by the enzyme Nis-P for the cleavage of the leader peptide, thereby releasing the active nisin molecule. Nis-EFG and Nis-I functions to protect the producer strain from the toxicity of its own bacteriocin. And lastly, Nis-K and Nis-R are histidine kinase and response regulator proteins that controls the transcription of these biosynthetic genes. In this regulatory system, the nisin molecule serves as the activator molecule of the histidine kinase Nis-K. So the phosphorylated Nis-K activates the response regulator Nis-R, which binds to the promoter sequence in the biosynthetic genes. In short, the presence of nisin molecules in the environment triggers the strain to produce more by the activation of its transcription. The NICE system exploits the quorum sensing based regulatory mechanism of nisin. So again, the actors of NICE system are first the nisin molecule, NISK, NISR, and the nisin promoter sequence. So in the dual-plasmid expression system that we constructed, we cloned the structural genes of Enterocin NKR-5-3B and LacticinQ under the nisin-inducible promoter p-Nis. So while the biosynthetic enzymes are encoded in a separate plasmid vector under the constitutive promoter p32. So when we transform these plasmid vectors into the host that carries the Nis, R, and K, I want to emphasize also that the origin of replication of these vectors, the ORI, operates through a different system so that it will not compete with the host machinery. So this pill-based plasmid has a rolling circle ORI, while the plasmid carrying the biosynthetic genes have a theta replication system. So once this strain is cultivated and an exogenous nisin A is added in the environment, even at very low concentration, it triggers the transcription of the genes upstream of this nisin promoter.
Our apologies, we are having some technical problems. Mr. Datul is already fixing it.
So for us to see the practical implication of this system, I want to bring your attention here in this picture below, because this is the reverse transcription PCR analysis of these genes, of these Bacteriocin structural genes. So in this reverse transcription analysis, what we did here is that we isolated the messenger transcript of these strains, and then we synthesized the complementary DNA. And using this cDNA as template for PCR, we can somehow estimate the level of transcription of these genes under different condition of different concentration of nisin. So as clearly shown here, that the level of transcription when nisin concentration is increased, there is an upregulation. But this upregulation was not observed in the control mRNAs, 16S RNAs of these strains. And now I want to bring your attention in this picture above, because this is the bioactivity of the strains under increasing concentration of nisin A. So here I want to emphasize that the nisin A that was used is well below the minimum inhibitory concentration of this indicator strain, because the MIC of this indicator strain is around 500 nanomolar, but the highest nisin added was only around 3 nanomolar. So here clearly, with the increasing concentration of nisin added in the agar loan, the size of the inhibition zone is increasing, suggesting that the amount of Bacteriocin for LacticinQ and Enterocin NKR-5-3B is increasing with the increased amount of nisin, which is a direct consequence of the increasing transcription of its genes. So the practical implication of this inducible bactericid production system is summarized in this table, because this is the these are the strains that carries the dual-plasmid expression system, and they are cultivated in different medium. So MRS and M17 represents the complex and expensive medium. TSBYE represents the simple and inexpensive medium. So here I want to bring your attention in this two strains that carries the structure genes under the nisin promoter, and the biosynthetic genes of the circular bacteriocin under the P32 promoters, and for LnqQ as well. What I want to show here is that when these strains are cultivated in expensive medium, and when nisin was added in induced condition, so non-induced meaning no nisin was added, in induced condition, a good amount of bacteriocins were produced, but no bacteriocin was produced when nisin molecule was not added. Now, but when the strain was cultivated in an inferior medium like TSBYE, although there was a good amount of there was bacteriocin activity detected, but it was relatively lower compared to that in complex medium. So the reason behind this, it is because the biosynthetic enzymes, which are encoded in a plasmid vector that is not under the nisin promoter, acted as the limiting factor in the processing of the precursor peptide. So in order to circumvent this problem, we cloned the nisin promoter sequence and replaced the P32 with the nisin promoter sequence, so that both the structure genes and the biosynthetic genes are now under the nisin promoter. So with that, even the strain was cultivated in simple and inexpensive medium, a very good amount of bacteriocins were produced. So these results clearly show that we can produce the circular bacteriocin and leaderless bacteriocin in simpler media, meaning the much cheaper media. Okay, so now let's move on to the next research project on our efforts to bioengineer the circular bacteriocin NKR-5-3B to enhance its bioactivity through NNK scanning. So we have previously solved the 3D NMR structure of this bacteriocin, and this is the skeletal backbone and side chain structure of the bacteriocin, and this is the cartoon representation of the helical structure of this molecule. And the NMR data suggests that around this region is highly dynamic, which gives the bacteriocin molecule its flexibility. And this suggests that this region play an important role in its bioactivity. And looking at the hydrophobic structure plot of this bacteriocin molecule, we found that the overall structure of this bacteriocin molecule encloses a tightly packed hydrophobic core with a lot of hydrophobic patches. So with that information, we wanted to create a mutant library by introducing point mutations at the residues that are likely to play an important role in its bioactivity. These mutants were obtained by PCR using degenerate primers, wherein the codon of each target residue was replaced with N and K, where N is a nucleotide and K is either G or T. So by doing so, the probability of the codons with all 20 amino acids are represented equally with a very low probability of the introduction of premature stop codons. So this is the mutant library that we have created of the residues V25 to V35, where this is the helix number 2. So here we summarize the mutations or the replaced amino acids according to their properties, and the figures in this panels represent the bioactivity relative to that of the wild type. If it's below 100, meaning the bioactivity decreased. If it's higher than 100, meaning the bioactivity of the resulting phenotype was enhanced relative to the wild type. And these are the mutant library at the flexible region or the Inter-Helix number 3 from V36 to A43. In all, we were able to obtain 13 mutant phenotypes that exhibited enhanced bioactivity relative to the wild type. The most notable phenotypes are V32C, V32A, and V32G, which showed 233, 217, and 183% enhancement of its bioactivity. We also compared the growth profiles of these Bacteriocin variant expressing mutants, and we were not able to detect any significant change in their growth profiles, suggesting that the immunity modules of the native form of this bacteriocin still functions to this derivatives or bacteriocin derivatives with enhanced bioactivity. But what is very surprising is that when we purified these mutant phenotypes or these bacteriocin derivatives, the elution time of these bacteriocins were significantly changed. So for instance, the V35N derivative was eluted around 17.567 minutes, but L40G was eluted at a much earlier time, at 12.567 minutes. Now, when we examine the molecular properties of these phenotypes, we were not able to see significant change in their isoelectric points except for V32C, but the hydrophobic index or the gravity index of these phenotypes or of these derivatives were significantly changed. We also checked if the introduced mutations altered the secondary structure orientation of these bacteriocin molecules, because it is well known that the secondary structure of peptides and proteins highly influence their bioactivity. But as shown here, the orientation of the alpha helices of the bacteriocin derivatives are the same as that of the native bacteriocin. And this supports the notion that the changes in the bioactivity of the bacteriocin derivatives were a result of the changes in the molecular properties of the variant rather than its structural conformation. So to sum up, bacteriocins are proven antimicrobial compounds, but its production has remained expensive. Circular and leaderless bacteriocins are exceptionally attractive antimicrobials, and for specific molecular strategies to improve their functionalities, we can use controlled functional expression such as NICETM, which can offer solution in lowering the cost of its production, but this requires detailed information of its biosynthetic machinery of every bacteriocin. And in order to enhance its potency, we can bioengineer the bacteriocin to hoping to obtain derivatives with more potent bioactivities. But again, this requires established heterologous expression system of every bacteriocin. So that's all. Thank you very much, and I'm looking forward to your questions, and I'm more than willing to entertain your questions. Thank you.